आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
We are immensely happy to receive a conversation from Dr. Utpal Nandi in this episode of podcast series. He will be introducing himself and will be sharing his academic journey with us.
Myself, Dr. Utpal Nandi, working at Bose Institute in the Department of Chemical Sciences. I completed my bachelor's, my master's and doctoral research from Jadavpur University, Department of Pharmaceutical Technology. After that, I joined Indian Pharmacopoeia Commission and worked there around 4 years at RST division. Then I moved to CSR Indian Institute of Integrative Medicine, Jammu. There I worked at Pharmacology division. Then I moved to Bose Institute and joined here as an associate professor.
Dr. Nandi now will be sharing his area of research in short.
My area of research is pharmacokinetics in drug discovery. Pharmacokinetics involve ADME PK. ADME in short, absorption, distribution, metabolism and excretion. What is its role in determining whether a drug will succeed or fail? PK helps us to understand how a drug moves through the body.
Dr. Nandi now explains his research work in details.
ADME stands for absorption, distribution, metabolism, elimination. Absorption means how well is the drug absorbed into the bloodstream. Drug have to be effectively absorbed into the systemic circulation. We evaluated some parameters which is related to druggability of the candidates. It may be physicochemical properties like solubility in water, simulated gastric fluid, simulated intestinal fluid, because poor water solubility is a great concern for a molecule to be developed as a drug. On the other hand, lipophilicity is another parameter which is important for drug discovery. Optimum log B and log D is crucial for a candidate. For example, propranolol versus sotalol. Propranolol is higher lipophilicity and therefore remain in the body for longer time. Ionization constant is an important factor where it can dictate about the absorption in which part of the gastrointestinal tract in case of oral absorption process. Chemical stability is one of the parameter which dictates about the absorption behavior of the candidate. For example, omeprazole is a drug which is degraded in the stomach. Therefore, in reality, we take omeprazole in the form of enteric coated granules so that it will open up in the when it reaches to the intestine. Some of the cases, drug absorption behavior depends on some efflux transporter activity like P-glycoprotein, BCRP transporter activity like that. D stands for distribution. Plasma stability is one of the parameters related to distribution. Some products are developed available in the market where the active molecule converted in the plasma after absorbing converted into the plasma to its active form. Plasma protein binding is not expected to be high, but it's not a constraint for a candidate to be developed because free fraction is important for the efficacy of a particular molecule. RBC partitioning is one of the parameters which have to be taken care during the development process, because if the molecules bind to RBC, then it may cause some toxic activity. Drain permeability, if I going to develop a candidate for CNS related drug discovery, then compound must have to be cross blood-brain barrier, which is important criteria for that molecule to be active. If I talk about metabolism part, then during the preclinical discovery phases, we can elucidate what is its metabolic stability using different liver microsomes. That may be different species to species variation can be determined. At the same time, what are the metabolites generated from the candidate can be elucidated, because some of the molecules developed and come into the market later on evaluated that metabolite is also active, like diazepam converted to oxazepam, later on oxazepam come into the market as a drug. Cytochrome P450 is one of the critical liver enzymes which is involved in the drug metabolism process. When two drugs administered simultaneously, there is a chance of drug interaction.
Dr. Nandi will be explaining his research work with examples.
If a drug of a particular CYP substrate is combined with another drug which is inhibitor of that particular CYP inhibitor, in that case drug interaction can happen. That may cause morbidity as well. On the other hand, in that case toxicity may also precipitate. For example, if rifampicin combined with bedaquiline for treating tuberculosis patient, in that case bedaquiline efficacy cannot be obtained, because rifampicin induce the metabolism of bedaquiline and plasma concentration will be decreased. If I talk about excretion, there are several routes of excretion of that particular candidate or drug, like urine, feces. Among this process, some of the studies, oral viability of a drug candidate is also important, because ultimately efficacy depends on what the plasma concentration of that particular candidate reaches to the bloodstream. At the same time, dose proportionality have also been determined, because up to which level there is a linearity or not, we have to be checked for oral route of administration. Some toxicokinetics evaluation can also be done to elucidate at higher dose level in which organ it will reach at higher amount. Disease state kinetic study can also be done to determine if any disease related factor change the pharmacokinetics of the drug compared to normal state. Some of the molecules, brain pharmacokinetics is also needed for some of the molecules where it is mandatory to reach the candidate to brain. If I co-administer two drugs simultaneously, then cytochrome P450 mediated drug interaction may occur. It may be CYP induction related, it may be CYP inhibition related. For example, if I administered isoniazid with phenytoin, then plasma concentration of phenytoin is going to increase. In that case, there is a chance of phenytoin toxicity precipitation. If I administered rifampicin with bedaquiline, in that case plasma exposure of bedaquiline is going to reduce and therapeutic efficacy of bedaquiline will be loose. If tetracycline is taken along with milk, then it forms a chelate, then it will not absorb. Therefore, tetracycline will not going to act. If I take a drug along with antacid, it may change the absorption process. Drug is going to absorb from stomach, and that is a acidic drug. If I take with antacid, then whole ionization process is changed. Therefore, absorption which is going to happen, that will be affected. Even extent of absorption will be affected more. In our lab, we have developed a battery of assays at the in vitro, in vivo, ex vivo, in situ level, so that quickly we can assess the druggability of the molecule.
Dr. Nandi now focuses on how is the research work currently happening in India and also speaks on the way forward.
Pharmacokinetics is the integral part of drug discovery process. Medicinal chemistry research is ongoing throughout the world. If I talk about Indian context, there is a huge chances of natural product driven drug discovery, in which area we are working for last several years. Therefore, Indian government formed some set of rules for IND application, which is already existing for new chemical entity, now promote in the form of phytopharmaceutical. That can also be treated as new chemical entity. Phytopharmaceutical is basically a extract which is later on enriched for certain markers. Here the markers are defined. That means you have to have 4 markers in that particular extract, and quantitatively that markers will be in particular defined range. We are giving efforts towards development of phytopharmaceutical because India have a vast natural resource, and important chemical scaffold from plant-based natural product are chemically modified for better druggability and better efficacy of a particular candidate. If I talk about drug discovery and development process, research are ongoing both academic as well as industry point of view. If I think about Indian context, in academia, there is scope of discovery process and then join hands with industry for further development process so that overall process can be accelerated for drug discovery and development.
AI, artificial intelligence, plays an important role in this research work. Dr. Nandi now speaks on that.
In the drug discovery process, medicinal chemists are already using in silico modeling. That means use several computational tools to optimize the candidate. Now AI is coming into the market where AI as well as machine learning are now playing major role in refining that predictions. Pharmacokinetics, that means ADME and PK, are fundamental pillar of drug discovery. A drug may look promising in the lab, but if it is not absorbed well, metabolized efficiently or cleared properly, it's destined to fail. Ultimately, early evaluation of pharmacokinetics of a candidate will be helpful to save money, to save time, as well as successful identification of a candidate into a marketed drug.
We are really thankful to Dr. Utpal Nandi for sharing his valuable research work with us. Thank you so much.